The second line of defense is more sophisticated than the first line of defense because the immune system should actually do something to deal with the pathogens that have penetrated the first line of defense. You want to imagine the second line of defense as being a combination of the FBI and the armed forces involved in combat or rescue. We're going to classify the second line of defense into three types: protective processes, protective proteins, and protective cells. So we'll start with the protective processes. There are two types of protective processes that we will look at. One of them is inflammation, and the second is fever. Both inflammation and fever are beneficial to the body unless they go out of control. The second is protective proteins. Let's look at two types of protective proteins. One of them is the interferon, which is an antiviral protein, which we will look at closely in a bit. The second is a complement protein. Complement proteins are a set of proteins floating around in the blood, waiting to be activated by the presence of a pathogen. The third group is the protective cells. We will take a look at two major protective cells at the second line of defense, and they are the natural killer cells and the phagocytes. The natural killer cell is a kind of lymphocyte, and the phagocyte is the macrophage or monocyte. Let's imagine that the skin has been penetrated by a sharp object, bringing with it some bacteria that we see in blue. The bacteria has now penetrated the epidermis of the skin and it is in the dermis. The injured tissue produces alarm chemicals shown in yellow, and these alarm chemicals stimulate a cell called a mast cell that is shown in blue. The mast cell is a cousin of the basophil, and we know that basophil releases histamine, and histamines make capillaries leaky. So that's what we see here. The histamine from the mast cell makes the capillaries leaky so that neutrophils and macrophages can squeeze through the capillary and arrive at the site of injury. So that's what we see here. The neutrophils and macrophages arriving at the sites of injury. So let's go over the steps one more time. The first step is that the tissue is injured by a sharp object and bacteria enters the skin and reaches the dermis. The injured tissue now produces a vast variety of chemicals, one of which stimulates the mast cell. So alarm chemicals are released by the injured tissue. The third step is that mast cells are stimulated. Mast cells are cousins of basophil. They are like the basophil, and they release histamine. And histamine is known to make capillaries leaky so that more things can come out of the capillary. Once the capillaries are made leaky, the white blood cells can now squeeze out of them. That's step 4. The neutrophils and the macrophages are attracted to the site of injury, and so they move to that area and then they squeeze out of the capillaries and arrive at the site of injury. Step 5 is that the neutrophils and macrophages kill the bacteria via phagocytosis. Fever is an indicator that there is an infection. However, fever is actually beneficial because it is a process that allows us to fight off infection. Our body temperature is set around 37 degrees Celsius. We call this the set point, and it is registered in the hypothalamus. Even though we say that 37 degrees is the set point, our body temperature tends to go a little bit higher and a little bit lower than 37 degrees. We call this an oscillation. We say that it oscillates around the set point. When there is an infection, however, the set point goes way up to 39 degrees Celsius. So now the hypothalamus registers the set point, new set point, at 39 degrees Celsius, and it stays up there until the infection is over. Once the pathogen numbers go down, the set point goes back to 37 degrees Celsius, and we return to the oscillating 37 degrees Celsius. Now let's look at these stages one by one. At the onset of an infection, certain lymphocytes release a chemical known as a pyrogen. Pyro means fire. So this pyrogen helps to raise the set point from 37 to 39 degrees Celsius. So the new set point is now 39 degrees Celsius. The body needs to now achieve that new set point, and while it does that, you're going to feel cold, shivery, and a little uncomfortable. Once we reach the set point, the new set point, which is 39 degrees Celsius, the body temperature will oscillate right around the 39 degrees Celsius. This continues until the pathogens have been destroyed. Number 4 represents the end of the infection. Once there is no more pathogen, there is no more pyrogen release from the lymphocytes, and so the set point goes back to normal. Let's recap these steps one by one. Number 1, there is an infection and there is a pyrogen secretion. 2 is the temperature set point going up. So it goes to a new set point. Stage 3 is the fever set point. We have arrived at the fever set point and you are maintaining this set point. 4, the infection has ended and the set point goes back down. Step 5 is the body temperature going back to normal, returning to the original set point. Now for the benefits of fever. Pathogens in general do not like high temperatures. They don't do very well at high temperatures. So fever helps to retard bacterial growth. It also slows down viral replication. The second benefit is that high temperatures raise our metabolic rate, and in doing so, they also intensify our immune reactions, so we are better able to fight off the pathogen. So fever can actually be a good thing.